Insulin is a hormone produced by the pancreas, which helps regulate blood glucose levels. This hormone is vital for the transport and storage of glucose in cells and helps to use glucose as a source of energy for the body. Insulin acts as a key to allow glucose to access cells. If glucose cannot enter the cells, it accumulates in the blood. The pancreas produces insulin in two different ways. A slow and continuous that helps glucose levels are always stable between 70 to 100 mg/dl, known as basal secretion. Another quick and in greater quantity secreted generally when the sanguineous glucose is in high values after the foods, well known like pulsatile secretion. Fast-acting or ultra-fast insulin. It is rapidly absorbed from the adipose, subcutaneous, tissue in the bloodstream. It is used to control blood glucose during the ingestion of food or beverages, and thus correct high blood sugar levels. Classification: human. For almost 50 years the only insulin available was that of cows and pigs, until Novo Nordisk discovered how to make synthetic human insulin using the key ingredient of a loaf of bread, yeast. Human insulin is obtained by genetic engineering to add the gene for the production of insulin to its DNA, that is, it has been genetically modified to produce insulin with a chemical structure identical to that produced by the human pancreas. Analogs. In recent years, so-called insulin analogs have been developed, which are molecules similar to human insulin, modified by genetic bioengineering in which the amino acid sequence has been changed. These insulins have pharmacokinetic characteristics that potentially allow improving the utilization profile of conventional insulins. Insulin analogs arise from biochemical modifications of human insulin. These modifications of the insulin molecule alter both the absorption and the onset and duration of the action, which offers advantages over conventional insulins. Currently, there are 3 insulin analogs with ultra-rapid action: insulin lispro, aspartic and pulsatile. There are also 3 long-acting analogs available: insulin glargine, detamir and alblin. In addition, they exist that reproduce better the physiological secretion than the regular human insulin and the neutral protein hagedorn, NPH, respectively. In this post we will focus on the 3 rapid-acting insulin analogs. Active principle. The active principle is defined as all matter, whatever its origin, human, animal, vegetable, chemical or otherwise, to which an appropriate activity is attributed to constitute a medicine. In the case of insulins, all are composed of an active ingredient which is called a trademark. Lispro, Humalog. The active ingredient lispro is a short-acting version of artificial human insulin. Insulin lispro-protein, or NPL, is an analog of human insulin in which it inverts the order of 2 amino acids, lysine and prolin, to which protein is also added, in order to delay the onset of action and prolong the duration of the effect. Mechanism of action. This begins to act between 5 and 15 minutes after its injection and remain active between 3 and 4 hours. The presentation of lispro is given in vials, or cartridges containing insulin, and placed in dispensing pens and dispensing pens containing cartridges such as pens. They are known for their trademark Humalog and sold in different formats. Aspart. Insulin aspart is a rapid-acting human insulin homolog, used to reduce high blood sugar levels, hyperglycemia, in adults, adolescents, and children 2 years and older with diabetes mellitus. It is obtained by genetic engineering, using a strain of Saccharomyces cerevisiae in which a modified gene of human insulin has been inserted. Insulin aspart only differs from human insulin in that the amino acid prolin at position 28 of the B chain has been replaced by an aspartic acid. This substitution reduces the tendency of the human insulin molecule to form hexamers, so it can be absorbed more quickly from the site of injection. Insulin aspart is classified in the category of ultra-fast insulins. Mechanism of action. Insulin aspart produces a faster onset of action than soluble human insulin, reducing postprandial glucose concentrations during the first 4 hours after a meal. The duration of action of insulin aspart longer than that of human insulin after subcutaneous injection. Glycine. Insulin glycina is the 3rd analog of human insulin of the fast action and short duration available in our country. It is obtained by recombinant DNA technology in strains of Escherichia coli. Mechanism of action. Rapid onset presentation, 5 to 15 minutes after subcutaneous administration. The maximum effect is reached at 55 minutes after the injection. The duration of the action is short, 2 to 5 hours. Regular. Regular insulin, sometimes indicated ordinary soluble insulin, or regular or unmodified insulin. Regular insulin rDNA is an insulin produced by a strain of Escherichia coli that has been genetically altered to produce human insulin. Regular insulin consists of insulins in crystals dissolved in a fluid for injection and is identical in all human insulin, without any structural modification or additions to modify its action. It is indicated in the treatment of diabetes mellitus, when insulin treatment is required. It is also indicated for the treatment of hyperglycemic coma and diabetic ketoacidosis, as well as for obtaining pre, intra and postoperative stabilization of patients with diabetes mellitus. Mechanism of action. Regular insulin has a fast effect and a short action, 4 to 12 hours, compared to other insulins.